In this subtopic, you will learn about development of pollen grain or male gametophyte and formation of male gamete, development of embryo sac or female gametophyte and formation of female gamete, and also double fertilization in the formation of seed. In flowering plant, the flower is the reproductive structure. So in the flower, there are petal, receptacle, and also the sepal. Other than that, a flower also has anther, filament, stigma, style, and the ovary. Inside the ovary is ovule. Anther and filament are the components of stamen. Stamen is male reproductive organ in flower. Anther is the site of pollen grain development, while filament hold the anther. Stigma, style, and ovary are components of carpel, which is the female reproductive organ in flower. Stigma is the landing site of pollen grain. Style will connecting stigma and ovary. Ovary is the site of embryo sac development. The development of pollen grain or male gametophyte is occur within anther. In each anther, there are 4 pollen sac or there are 4 microsporangium. So this is the microsporangium or pollen sac. Within each microsporangium or pollen sac, there are many diploid cells known as microsporocyte or microspore mother cell. Each of these diploid microsporocyte undergoes meiosis to produce 4 haploid microspores. So this is the 4 haploid microspores, which is also known as tetrad cell. Each of the microspore will undergoes mitosis to produce haploid tube cell and haploid generative cell. These two cells, tube cell and generative cell, is found within male gametophyte or pollen grain. So pollen grain also known as male gametophyte. Later on, when the pollen grain land on the stigma, the pollen grain will become mature where the generative cell will undergo mitosis to produce 2 sperms or 2 male gametes. The tube cell will produce pollen tube. This is the structure of pollen grain. Pollen grain is protected by a thick outer sculptured wall known as axine. The surface of the pollen grain is rough because it helps the pollen grain to land and attach on the stigma. Development of embryo sac or female gametophyte is occur within an ovary. Inside ovary is an ovule. Inside ovule is a megasporangium, and inside megasporangium is a large diploid cell known as megasporocyte. The megasporangium is protected by integument with one end known as micropyle. Within megasporangium of ovule is a large diploid cell known as megasporocyte or megaspore mother cell. The megasporocyte or the megaspore mother cell undergoes meiosis to produce 4 megaspores, which are haploid. This division involving unequal division of cytoplasm, where only one haploid megaspore receiving a large volume of cytoplasm, which later on become surviving megaspore. The other 3 haploid megaspore receive less cytoplasm volume and will be degenerate. So from 4 megaspores, only one will become survived and the other 3 will be degenerates. The surviving megaspore will undergo mitosis 3 times, producing 8 nuclei or 8 nucleus, where later on the 8 nuclei will develop into 3 antipodal cells, 2 polar nuclei, 1 egg cell, and 2 synergid cells. So 3 antipodal cells, 2 polar nuclei, 2 synergid cell, and 1 egg cell. All of this is located within embryo sac or female gametophyte. So this is the female gametophyte with 8 nucleated different stages of cell. This is a structure of embryo sac after the megaspore undergoes 3 times mitosis. Near the micropyle end are 2 synergid cells with an egg cell. At the opposite of micropyle end are 3 antipodal cells, and at the middle of the embryo sac is a central cell containing 2 polar nuclei. In flowering plant, fertilization occurs twice, where the first fertilization is fusion between one haploid sperm with one haploid egg to form a diploid zygote, and second fertilization is between one haploid sperm with 2 haploid polar nuclei to form a triploid endosperm. Hence, the process of fertilization is double fertilization, and the product is seed. So, what happened during fertilization? Firstly, the pollen grain will land on the stigma by the process of pollination. So, pollination is process transfer of pollen grain from anther to the stigma. So, once pollen grain lands on the stigma, the pollen grain will absorb the moist and nutrient on the stigma. Stigma has a moist and sticky surface because it is rich with nutrients. So the pollen grain will germinate. When the pollen grain germinates, the tube cell will produce pollen tube. And inside the pollen tube, the generative cell will undergo mitosis to produce 2 haploid sperms. Tube cell form pollen tube. In pollen tube, generative cells form 2 haploid sperms by mitosis. The pollen tube will elongate along the style towards the ovary, and then the pollen tube will fuse with the embryo sac through the micropyle, the opening of the ovule. The synergid cell inside the embryo sac will attract and guide the pollen tube towards the embryo sac. Pollen tube fuse with embryo sac via micropyle and guided by synergid cells. When the pollen tube fuses with the embryo sac, the pollen tube will discharge the 2 haploid sperms into the embryo sac. One of the sperm will fuse with the egg cell to form diploid zygote, and the other sperm will fuse with the 2 polar nuclei to produce a triploid endosperm. So, first fertilization, fusion between one sperm and one egg cell to produce diploid zygote. Second fertilization is fusion between one sperm with 2 polar nuclei, producing triploid endosperm. The zygote will develop into embryo. The endosperm will provide nutrients for the embryo development. The ovule or embryo sac will develop into seed, and the integument that protects the ovule will develop into seed coat.